morning everyone. As we all know, today I am with another topic, that is dividing phase, otherwise called as M phase or mitotic phase. This phase is actual cell division on basis of in which cell it is occurring. It can be divided into two types, that is mitosis takes place in somatic cells as well as meiosis takes place in germ cells. As of now, let us talk about mitosis. First, we need to understand of mitosis. Mito means thread, osis means state. In 1882, Fleming given the word, the term mitosis. In general, this mitosis takes place in somatic cell. That's why it is called as somatic division. At times, it also takes place in the germinal cell during the multiplication phase. This is exception, no? As a result of this mitotic division, we'll have two daughter cells along with same chromosome number, which is diploid, n plus n to n. At times it takes place in haploid cells also. Will get the two daughter cell as the top parent cell. Since having the same number of chromosomes, that is also called as equational division. In general, this mitosis takes place in somatic cell. That's why it is called as somatic division. At times, it also takes place in the germinal cell during the multiplication phase. This is exception, no? As a result of this mitotic division, we'll have two daughter cells along with same chromosome number, which is diploid. At times it takes place in haploid cells also. Will get the two daughter cell as the top parent cell. Since having the same number of chromosomes, that is also called as equational division. Fundamentally, it is related to the growth of the individual from zygote to adult stage. Now we'll see the stages of the mitosis. First one is karyokinesis and cytokinesis. Karyokinesis means nuclear division. Cytokinesis means division of cytoplasm. So, some cells undergo the karyokinesis but not cytokinesis. Results in a cell multiple nuclei is known as pseudium. It is basically multiplication of cell number during the embryogenesis and blastogenesis. Fundamentally, it is related to the growth of the individual from zygote to adult stage. One of the basic characteristics of the mitotic cell division, which is meant for growth due to multiplication, is that it gives rise to two daughter cells which are resemble to each other, and so the parent cell quantitatively and qualitatively. The basic outline of mitosis is same in all organisms. Basic outline remains the same in all individuals. As a process, mitosis is similar in all animals and plants. Mitosis starts at the culmination point of interphase. It is a short period of chromosome condensation, segregation, and cell division. Mitosis is important for the replacement of cell lost to natural friction, wear and tear, and wound healing. Mitotic cell division is mitotic cycle is divided into 5 subphases: prophase, prometaphase, metaphase, anaphase, and telophase. Prophase, prometaphase, metaphase, anaphase, and telophase. These are 5 substages. Firstly, we can see the prophase. This is the longest phase of karyokinesis. Appearance of thin thread-like condensed chromosomes marks the first phase of mitosis. Is called prophase. The cell becomes spheroid and more refractile and viscous. Each prophase chromosome composed of 2 filament-like structures is known as chromatids, which are held together by a centromere. Where they held together, this is called centromere. Over that, this is cap-like. These cap-like structures is known as kinetochore. As prophase progresses, they become thick and shorter. They are held together by a centromere, special DNA containing region that is called centromere. As prophase progresses, it becomes shorter and thicker, and they are special DNA structure containing which are held together by one to another is known as centromere, and a cap-like structure which are known as kinetochore. This centromere, otherwise called as primary constriction. During the elderly stage of the prophase, they are evenly distributed in the nuclear envelope. During the LD prophase, the centromeres with the chromatids which are held together, they are evenly distributed the nuclear cavity. When they start deposing or organizing, they are vacate the empty space at the central nuclear cavity. Gradually, they are moving towards the poles, northern and southern. In elderly prophase, the chromosomes appear like a ball of wool. It's known as sperm. Cells, centrioles formed and moving towards the either poles. In center, counterparts are called as spindles in animals. However, in plant cells, no need of centrioles. Without centrioles only, spindle fiber formation takes place in plant cells. That is the difference between the animal cells and plant cell. So these spindle fibers are made up of 3% of RNA, 97% of tubulin, which are made up of spindle fibers. These spindle fibers are organized with the help of calcium bound proteins is known as calmodulin. Calmodulin in plant cell and animal cells. The diameter of the spindle fibers are maximum at the equator. These spindle fibers have positive end towards the equator, negative end towards the pole, which microtubule towards the pole radiating around the centriole is known as aster microtubule, which are towards the pole, it is known as polar microtubules. Next, which are attached to the kinetochore. So that is after centromere there is a one cap-like structure, no, that is kinetochore. This microtubule kinetochore, which microtubule is attached to the kinetochore, is known as kinetochore microtubule. So some microtubules are attached to the kinetochore with the chromatids where it is held together centromere. That microtubule is called as discontinuous, means it is not continued to here, one pole to other pole. Some microtubules are one certain pole to northern pole to southern pole, it continuously spreading. So this is called as continuous microtubule. Astus, centrioles, microtubule, all together called as mitotic apparatus. Nuclei and nuclear envelope no longer visible because it is a late prophase, no? All going to disappear, and Golgi complex and endoplasmic reticulum also start disappearing in this late prophase. What is the difference between the LD prophase and late prophase? LD prophase, the nuclear chromosomes look like a ball-like structure, that is P-rem stage. Here all going to disappear and at the center empty space will be. This is the late prophase. Pro-metaphase is transitionary between prophase and metaphase. Each one surrounded by so-called aster. Which is composed of the microtubule radiating all directions. Just as centromere tend to protect the minus end from the depolymerizing, which is composed of microtubules in all directions. Two pairs of centrioles. Two pairs of the centrioles along with the asters move opposite poles of the cell become situated in antipodal positions. Between separating centrioles form spindle. The microtubule of the spindle arranged like a cones base to base, like this, narrowing a point towards the pole or end, broad at the center. In other way, can say there are mitotic spindles are three main types. One is polar fibers, which extend two poles of the spindle towards the equator. These are, means from here to here, these are called polar fibers. Second one, kinetochore, which attach to the kinetochore of centromere of the mitotic chromosomes. Third one, astal fibers, radiate outward from the poles towards the periphery in cells of most plants. However, spindle fibers are formed without centriole and lacks the aster. Lastly, during the prophase, nucleolus gradually disappears. Disintegration, disappearance of the nuclear envelope marks the end of the prophase. The process incompletely understood, however, the following two factors may be involved in this process. What are those? We'll see. What are those? First one, enzymatic. Enzymatic action. Second one, physical action. Physical action. Enzymatic action either by some mitochondrial enzymes or cytosolic MPF mitosis promoting factor kinase or nuclear RNA. Nuclear RNA or ribosome. Physical action which physical stress exerted by the microtubule which become attached to the nuclear envelope.